So you might be wondering why I say that there are 2 genomes and 3 systems. And the reason that I say that there are 2 genomes and 3 systems is because we all have in the nucleus of each cell a genome. And we refer to this as our nuclear genome. And this is where the chromosomes are in the nucleus. But we also, outside of the nucleus, but inside the cell still, inside the cytoplasm, we have the mitochondrial DNA, or the mitochondrial genome. So we all probably have heard that the mitochondria are the powerhouses of the cell. Mitochondria are where cellular respiration goes on, and we'll talk a little bit about this later. But inside each cell, we have hundreds to thousands of mitochondria, and each one has a genome. And inside each mitochondria, there might be multiple mitochondrial genomes. And the mitochondrial genome has been extremely useful in forensics and in evolutionary studies, also in ancient DNA. The mitochondrial genome is much smaller. It's only it's a little more than 16,000 bases long. And it's double-stranded DNA, so it's complementary. But it's not paired the way that the chromosomes are. And so we've sequenced thousands of mitochondrial genomes, and there are multiple databases where we can compare our sequence of interest to these databases, and we can trace lineages, and we can all multitude of applications, but we will come back to that. So anyway, so inside the nucleus of each cell, we have the nuclear DNA, and this is what makes up the paired chromosomes. In the cytoplasm, outside of the nucleus, we have mitochondria, and inside each mitochondria, we have the mitochondrial DNA. In regards to the 3 systems, I say this because we have chromosomes, and of course you have maternal and paternal contribution of chromosomes, and then we have the mitochondrial DNA, or we annotate it as mtDNA. And the interesting thing about mitochondrial DNA is that there's only maternal contribution. So you don't inherit any mitochondrial DNA from your father. So all the children of your mother have the same, they've inherited their mitochondrial DNA from mom, and it's essentially clonal, which means that that the copy that each child or each offspring has is almost exactly the same as what mom has. There's no recombination, so it's essentially clonal. There are some instances where you do have paternal contribution. It's very rare, and it results in a severe genetic muscular disorder. So only maternal contribution for mitochondrial DNA. And then we have, going back to nuclear DNA, we have Y chromosomes. And of course the Y chromosome is passed from father to son. Females are XX chromosome, so have an X chromosome from mom and an X chromosome from dad. And male offspring have an X chromosome from mom and a Y chromosome from dad. And so in terms of when we're studying, if we're thinking in an evolutionary context, and we're thinking about how we study evolution and how we study how people are related to each other, how different populations are related to each other, and even how we are related to other organisms, we can look at nuclear. Oops, sorry about that. We can look at nuclear DNA, and so then that gives us information about maternal and paternal lineages and contributions. We can look at mitochondrial DNA variation, which is going to tell us only about the maternal lineages. But we can also look at Y chromosome variation, which is going to tell us about paternal contribution. And what we'll find, what we'll talk about a little bit later in the quarter, and for those of you who explore biological anthropology further by taking additional courses, you might take some courses where we talk about how do we use DNA to answer anthropological questions? Can we look at DNA variation and how it relates to and informs us about cultural practices? So that is why I say 2 genomes, 3 systems. You'll notice on this slide that I say that purines are double-ringed and pyrimidines are single-ringed. And when we're talking about DNA, purines are adenine and guanine. Pyrimidines are cytosine and thymine. In RNA, we have uracil rather than thymine, so uracil is a pyrimidine. And if you look at the diagram, you'll see that the molecular structure, this is what single-ringed versus double-ringed is referring to. And the pyrimidines are single-ringed, and the purines are double-ringed. And this is what contributes to the shape and the twisting of the double-stranded DNA so that we get the helix. It also contributes to base pairing and recognition of mispairing. Both DNA and RNA are made up of individual blocks that join together. The blocks are a nucleotide. Each nucleotide is made up of a base, a sugar, and a phosphate group. DNA stands for deoxyribonucleic acid. Deoxyribose is asymmetrical. One question that I'm asked often, because I'm in clinical molecular diagnostics, is this idea of if you have an allele that's associated with a disease, will you always get the disease? Most diseases and most of our phenotype, most traits that are expressed that make up the entire that contribute to making up the entire organism, most of these traits are complex, so multiple genes are involved. So at the molecular level, multiple genes interacting to produce a trait, a disease, a characteristic. There are disorders out there. There are diseases out there that are what we would call a simple Mendelian disorder, meaning that there's a substitution in that gene, then the individual is affected or expresses that trait. One gene, one phenotype. But really, most of our traits, our characteristics, most disorders are complex and multigenic. And we'll come back to this and talk more about this. Transcription is where the DNA that makes up a gene is read and copied into RNA. It's transcribed into an RNA strand, which will then ultimately be read or translated into a protein. So the best way to think about DNA and genes and chromosomes is a nested approach and also a systems approach. So if you look at each individual as a complete system, you see the whole of the individual. But if we were to look more closely, then we would see that each individual is made up of tissues, which make up organs. And the tissues are made up of cells. And if we'd look even closer and look at the cells, we'd see that each cell has a cell membrane, which is a lipid bilayer, and then inside that lipid bilayer, that cell membrane, we'd see cytoplasm, and we'd see organelles in the cytoplasm, and we'd see inside that cytoplasm, we'd see a nucleus, and the nucleus is enclosed by a nuclear membrane. Inside that nucleus are the paired chromosomes. The chromosomes sorry about that. The chromosomes are made up of DNA, strands of DNA. Genes are a segment of DNA on a chromosome that codes for something, contains the information that codes for the proteins that make up the organism. So transcription, or the process of reading DNA, or the gene, the sequence of DNA within the gene, to become RNA, to be transcribed into a messenger RNA, which ultimately will then be translated into a protein, has a direction to it. It always goes one direction. There's a start and an end, and it goes one direction. So it proceeds 5' to 3'. And on the 5' end, because that's the start end, the starting end, there's an initiation sequence. And this initiation sequence is a sequence that says, hey, this is where you start. It also regulates transcription. And then, of course, at the end, there's a codon, 3 bases, that says stop transcribing. This is the end of the gene. Translation is that process of making a protein, translating the messenger RNA into a protein. So the messenger RNA is the RNA that started out in the nucleus. It's the RNA that was the result of transcription of the DNA, of the gene. The pre-messenger RNA, or the immature mRNA, has the exons and the introns. Then the introns are cut out, the exons are spliced back together. That mature mRNA moves out into the cytoplasm, and ribosomes attach, and the ribosome is the organelle where the messenger RNA is translated into the protein. So you have this mature messenger RNA, and then it's going to be read and translated into a protein. Transfer RNA is a specific type of RNA that's going to bring that amino acid to the ribosome, and the amino acids, of course, are the building blocks of proteins. And so you have your messenger RNA, and the codons, which is that series of 3 bases, they're right next to each other, and the sequence of those 3 bases codes for a specific amino acid. The transfer RNA brings that amino acid and adds it into a growing chain of amino acids. What you'll notice in this slide is that the genetic code is redundant. And what this means is that there are 20 amino acids, but there are 4 base pairs. And each codon is comprised of 3 amino acids. So there are multiple combinations that code for an amino acid. So for example, if we look at this table and we look at, let's look at glycine. And you notice that glycine, so in the lower right corner, gly, G-L-Y, the amino acid gly is coded for by GGU, GGC, GGA, and GGG. So it's redundant. Somatic cells is referring to the cells that make up the tissues and organs of the bodies. And as we age, we all accumulate spontaneous random mutations in the DNA of our somatic cells. And these can contribute to disease. These contribute to cancers. But in terms of evolutionary studies, what we're most interested in is mutation in the DNA of germline cells. These are the eggs and the sperm.